Okay, so now we'll look at red blood cells. A couple interesting things about red blood cells. Mature red blood cells lack a nucleus. So as we kind of saw previously when we looked at hematopoiesis, as the red blood cells go and mature, notice we lose the nucleus right here before they are sent into the blood. So they are what we call anucleate. They don't have any nucleus, they don't have any organelles, they have no mitochondria, they have no endoplasmic reticulum, and they have a few enzymes for glycolysis, so they are good at anaerobic metabolism, and that's about it. What we've done, though, is we have taken all those materials out and shoved as much hemoglobin as we can inside. We'll talk about hemoglobin in a minute. A couple other properties of red blood cells: they tend to be very flexible. That's because they can twist and squeeze and move through very thin diameter capillaries. They can have osmosis-induced changes, so they can fill with water, as we kind of saw in lab with hypo and hyper and isotonic solutions. You can see here in the pictures they are biconcave discs, so they have this circle in the middle right here. That's a depression. It increases the surface area that allows better exchange of oxygen. And really their big job, what they do, is they carry O2, oxygen. That's their main job. And as I mentioned before, a reticulocyte is a immature red blood cell with a nucleus. And when they look at blood labs, sometimes they'll look at the reticulocytes you have. And if you have some sort of bone marrow problem where you're overproducing too many red cells, then you'll have what's called reticulocytosis, excessive number of reticulocytes in the bloodstream. As I mentioned on the previous slide, red blood cells have a biconcavity. And again, this depression right here, right, in this area right here, increases the surface area, which allows more oxygen, because that's their job, is to carry oxygen. Don't worry too much about what makes that up. Certainly it's, protein elements that help to hold the cytoskeleton elements that help to hold the red blood cell in its shape. But the biconcave disc, again, increases surface area, facilitates diffusion. Now, we just said that we basically, when we mature a red blood cell, it loses its nucleus and most of the other organelles. And what we do is we take the open space now and literally shove as much hemoglobin into a red blood cell as we can. So hemo, again, refers to blood. Globin is protein, so it literally means blood protein. It's a tetramer. It's got 4 subunits that make it up. And the key is each hemoglobin subunit has a little heme or iron portion. Okay? And these iron portions right here, this is where the oxygen binds to. So every hemoglobin molecule has the ability to carry 4 oxygens. Okay? So those 4 oxygens, right? Okay, that seems okay. And I think most people seem to remember that from anatomy. But what most people don't remember, or maybe just didn't get, pounded into your head, is that, I don't want to go closer. The pounded in your head is that on an average red blood cell, there are 280 million hemoglobin molecules. Okay, so 280 million. So these little tiny proteins basically shoved into these little tiny red cells, right? So let's kind of look at some stuff we learned before and kind of tie it together. So each hemoglobin molecule has 4 heme sites, right, that can carry oxygen. There are 280 million hemoglobin molecules in a red cell. There are 5 million red cells in a microliter of blood. By definition, there are 1 million microliters of blood in a liter, and the average human has 5.5 liters of blood. So that's like a gazillion oxygen molecules we can carry in our body. It's just amazingly big number if you think about it. A couple other things that come with hemoglobin then. We measure it like other proteins in grams per deciliter, and a normal hemoglobin is 14 grams per deciliter. For most people, once you get to 12 or below, you have anemia. Again, we have 5 million red blood cells within a microliter of blood, and we have 5 million microliters of blood within a human. So it's some pretty big numbers. Hemoglobin, besides carrying oxygen, it also acts as a buffer. So it helps to resist changes in pH, and that's because it can also bind the protein portion, the amino portion, actually, can bind the free hydrogen. And so that will help, because remember, pH is a measure of the free hydrogen. If the hydrogen is complex to something, it won't be free. So that's kind of how hemoglobin works. Now, in order to have sufficient hemoglobin, you need that raw material. You need iron. You also need the amino acids to make up the protein. But iron is required for the synthesis and function of hemoglobin. Now, iron, remember, has a chemical symbol Fe, and again, it's the oxygen that binds to the iron that allows us to carry oxygen in our blood. Generally, it's obtained by our diet. We recycle it fairly vigorously in our body because it's hard to get in our diet. If you have insufficient amount of iron, then you're going to not properly make hemoglobin, which means you're not properly going to make red blood cells. And so if you have the inability to carry oxygen, that's a disease called anemia. And there are many causes of anemia, and we'll kind of look at them a little bit later. And I want to look at the sort of the problem, dysfunctional things that have kind of to do with blood. Now, hemoglobin can carry several different molecules on it, not just oxygen, and this is where it becomes a little bit of an issue sometimes. So when oxygen binds to the heme portion of hemoglobin, it becomes oxidized. It's not any different in terms of the chemical reaction that happens when you leave a nail out in the backyard and over time it rusts. Okay, so that's the oxidation. And because the iron is becoming oxidated, oxyhemoglobin, that's heme that has oxygen bound to it, has the color red. Okay, so oxyhemoglobin is red. That's what gives blood its red color. Now, if we're not carrying oxygen on hemoglobin, then the iron is reduced. Okay, I'm not too worried about the oxidized versus a reduced section, but know then that if we're not carrying oxygen, we call that deoxyhemoglobin, and that turns blue. Okay, so blue. We tend to have more oxygen in our arteries than our veins because our cells use them. Okay, but that's not the only thing that we can carry on hemoglobin. Hemoglobin also carries some CO2. While hemoglobin carries about 97% of our oxygen, it only carries about 23% of our CO2. So it still carries CO2. Now, what's interesting is, unlike oxygen that is carried on the heme portion, the carbon dioxide, CO2, is not carried on the heme portion. It's actually carried on the amino acid. And that gives us a different name for it then. So the name for that is carb, which refers to CO2 actually. Amino, which means that the CO2 has been carried on the amino acid, not the heme. And then hemoglobin. So carbaminohemoglobin is what you call hemoglobin that's carrying CO2. All right. One thing that I forgot to mention, so let's just go back really quick. When we talk about deoxyhemoglobin, when there's no oxygen there, it'll turn blue. And one of the names for blue is cyan. And if you see somebody that has cyanosis, it means there's oxygen not going to a certain part of your body. So you could see it in someone who is cold and their lips will turn blue, someone who has extreme circulation problems, their fingers might turn blue, or someone who has cardiac arrest, you might see bluing of things. And you see that word oftentimes on medical shows where they talk about cyanosis, or they're cyanotic. It means they're not getting oxygen. So what's also really dangerous is the next one, where the CO, so this is carbon monoxide, right? So CO equals carbon monoxide. And carbon monoxide is being recognized as being more and more dangerous. In California now, for new construction, you must have a carbon monoxide detector in the house. And even when you rent houses online from like VRBO, they now will send you a thing saying, are you sure you want to rent? There's no carbon monoxide detector within the house. And in California, it's not a big deal typically. But what it comes from is it comes from the incomplete combustion of materials. And so carbon monoxide poisoning can happen if carbon monoxide gets too high, and this is the reason why. So carbon monoxide will also bind to the heme group. Okay, so carbon monoxide actually inhibits the binding of oxygen. There's only one spot to bind on the heme, and if carbon monoxide binds, then we can't bind oxygen. And so it's basically a competitive inhibitor of oxygen. Now, one of the problems with that is it has a really high affinity, and it's about a 200 times affinity for heme than oxygen does. So it means, kind of to put it a little different way, is if we had 200 oxygen molecules and 200 carbon monoxide molecules and 50 hemoglobin molecules. I say 50 because each hemoglobin molecule would have 4 heme sites on it. So that would be able to bind 200 of things, whatever they are. And so on average, if we had those 200 hemoglobin sites, 199 would bind carbon monoxide and only 1 would bind oxygen. Okay? So when carbon monoxide binds, we call that carboxyhemoglobin. Right? So again, the carb refers to the carbon dioxide or carbon part, oxy, right? That's the carbon monoxide, hemoglobin. It's not hemoglobin. So don't confuse that with carbaminohemoglobin. Carbamino is when CO2 binds, so carbaminohemoglobin. Carboxy is when carbon monoxide binds. And, it becomes very dangerous. The way we treat carbon monoxide poisoning is you put people in a hyperbaric chamber. You increase the pressure. Let me do it again. So there's an arrow up, and I'm just going to put P for pressure. And by increasing the pressure, you increase the partial pressure of oxygen, which basically increases its concentration. So remember that we said earlier this semester, if you have a low affinity for a receptor, you can make up for that by increasing the concentration. So that goes back to that idea of, again, receptor properties with saturation, competition, and specificity. And the competition part here is, even though carbon monoxide has this high affinity for the hemoglobin molecule, we can, through hyperbaric chamber, giving people high oxygen and increasing the pressure, we can actually replace the carbon monoxide with oxygen and get somebody back to normal. Now, we mentioned that iron was important in terms of the diet, and hemoglobin, the components of it are actually recycled, and some of it is considered waste and secreted. So your old red blood cells are removed by macrophages. And now macrophages are specific white blood cells that are very important in terms of destroying things. We have them in various parts of the body. As an example, we have what are called Kupffer cells. Those are in the liver and spleen, and what the Kupffer cells do is they remove old red blood cells. And the way they can tell they're old is they, just like many of us, they become less flexible as they age. And so what happens is these macrophages pick up the Kupffer cells and then destroy them. Now, what we do is we recycle many of the parts, not all of it, but the protein is broken down back into amino acids and goes back to the amino acid pool, the general pool of the body. So that's fine. And the iron, right, goes back to the bone marrow, becomes heme, and is recycled. And now we make more hemoglobin to stick in a red blood cell. But one portion of the heme, bilirubin, will be secreted. And so there is some part of bilirubin that is secreted. If we can't break it down and secrete it, then it builds up in the blood. And if it does that, then we get a condition called jaundice. And I think most of you are familiar with that condition. You know, here's a nice, on the left, a nice pink healthy baby. And on the right is a baby with severe jaundice. So you can see the yellow and things like that. And that's because of the bilirubin that we see. Now, in babies, jaundice is a little more dangerous because the bilirubin in an adult cannot cross the blood-brain barrier. But in a baby, it can because they don't have a complete blood-brain barrier. And so it can cause some damage on especially the brain, and can result in, deficits in terms of learning and development and things like that. So you don't like to have high bilirubin levels for very long. Now, in an adult, it's not a huge deal, to be honest, except that if someone has jaundice, it means it's a condition that's caused by some other problem. And that other problem could be really big and typically affects the liver. So it could be cirrhosis or cancer or hepatitis or some other thing. You see jaundice very, very common in babies, and especially firstborn premature male infants that end up getting jaundice. And it's again because they don't have a fully functional liver yet, so that liver that normally processes the bilirubin for secretion doesn't work very well, so the bilirubin builds up in the blood. Now, we said earlier that the red blood cell's primary function is to carry oxygen. So that is the most important thing. So when we carry the oxygen, how much oxygen we can carry is determined by several factors. And so let's kind of look at some of the different numbers. These are numbers you should know. There are different ranges for men and women. Typically, men tend to be on the higher end and women tend to be on the lower end. But these numbers make it pretty easy to remember. So these are ones you probably should have an idea of for each one. So how much oxygen we can carry is influenced by what we call the red blood cell count, or sometimes known as the total red cell count. And again, the average person has about 5 million red blood cells per microliter of blood. The other way to write that, and you'll see it oftentimes medically, is in cubic millimeters, right? So it's like milliliters is cubic centimeters, then microliters is cubic millimeters. So if we look at a red cell count, most people are going to hover around that 5 million. Men tend to be a little under or significantly over, like 6 point something million, 6.2, 4 million. Women tend to be significantly under, like 3.5 million to 5.4 million or something, but they both cross that 5 million mark. For hemoglobin, mentioned earlier that 14 grams per deciliter was normal. Again, men tend to have more hemoglobin, women a little lower. And 12 or below was a danger sign where it got too low. Also, in terms of hemoglobin, remember we have 280 million hemoglobin molecules within a cell. When we looked at hematocrit earlier, that was the packed red cell volume. The magic number is 42 for that. As a matter of fact, if you look at what is often called the hematocrit to hemoglobin ratio, hemoglobin is abbreviated HB and hematocrit is HCT, it's equal to about 3-to-1. So what that means is that normally if your hematocrit value, let's say to make math easy, because we can see it right here, is 42, then your hemoglobin is going to be one part of that. So you just divide 42 by 3 and you get 14. So kind of looking at how this ratio is important is in a normal person with normal physiology, it holds up pretty good. As a matter of fact, up until 30 years ago, we didn't bother to measure the hemoglobin in a blood test unless it was specifically asked. And we just literally estimated it using this formula. So as an example, if someone had a hematocrit of 30, their hemoglobin would likely be 10. Right? Hematocrit of 30 would be pretty low, and in fact, medically, anemia is considered to be a hematocrit that is 37 or below. Now, the ability of hemoglobin to carry oxygen is very important from a number of different standpoints. This is called the oxyhemoglobin curve, and we're not going to talk about it really. But basically what it shows is that how much oxygen we can carry in our blood. But what I'm really interested in this is what's the idea of percent saturation. Percent saturation is basically a measurement of how many sites are occupied with oxygen by the total number. So it's the number there it's the number over total. So as an example, to make the math really easy, if we just looked at one hemoglobin molecule, which has 4 heme sites, and it was carrying 2 oxygens, the percent saturation would be 2 out of 4, 50%. Okay? When we look at percent saturation, we can measure it with what's called a pulse oximeter. You see those in hospital situations where someone will be wearing one on their fingertip as an example, and it would show you the percent saturation. When blood comes out of the lungs and into the arterial tree, it's about 99% saturated, which means that 99 out of every 100 hemoglobin sites are carrying oxygen. By the time it gets back after circulating through the body to the lungs, it's only about 70% saturated. So that difference is the oxygen that was picked up and used by the tissues. I'm going to erase this so we can see this bottom part. Humans can tolerate down to about 30% saturation for a very brief amount of time, but what's actually needed for life in terms of how things work is, I just covered it, %50, right? So 5 is what's needed for life. So in order to maintain life, you need 50%. Now, when we look at the reduced ability to carry oxygen in blood, that's a disease called anemia. It's a bad name. Anemia literally means without blood, but it just means that you have a compromised ability to carry oxygen. So you don't carry as much O2 as you can, or you don't carry as effectively. The indicators that can cause that are, low red blood cell, low hemoglobin, or low hematocrit. So, it could be a combination of these or an individual one. It could be a reduced red blood cell number. If you only have half as many red cells, you're only going to carry half as much oxygen. It could be insufficient or too little hemoglobin. Again, if you're supposed to have 280 million hemoglobin molecules in a red cell, you only have half of that, 140 million, going to carry half as much oxygen. Or it could be a problem where you have an inadequate hemoglobin, a quality problem like sickle cell anemia or thalassemias, where there's a genetic defect that hemoglobin doesn't effectively carry oxygen as well as it should. Or it could be a combination of these things that can cause anemia. So looking at some of those in a little more detail, looking at the reduced number of red blood cells can come from a number of different factors. One, you could have normal size red blood cell, right? But hemoglobin would be less. Or you could have red blood cells that are too small or too big or other conditions, right? So it could be a production problem. Oftentimes production problems can be DNA or RNA issues. We need certain things in our diet to make red blood cells. Iron, because that makes up the heme portion. Vitamin B12, remember we said we absorbed that earlier in the semester with intrinsic factor, and that's important. And then folic acid, all are important in terms of making red blood cells. It could be that you have some sort of bone marrow disease where you don't make enough red blood cells in your red bone marrow. Or it could be you have inadequate production of erythropoietin. Maybe the cells that make EPO in your kidney aren't functioning. So we could have a decreased production. Most people lose sight of the other side of this, but it could be you become anemic because you have an increased destruction or loss of red blood cells. There is a disease called hereditary spherocytosis, which means you have weakened red blood cells, which get destroyed easier. Blood loss, menstruation. If you have a bleeding ulcer or a bad case of hemorrhoids, where you have a blood loss of 1 ml a day in a month period, you're probably become anemic. People who have mechanical heart valves, when they have heart valve replacement, also end up destroying their cells. If it's not a reduced red blood cell number, it could be an insufficient hemoglobin, or you just have an iron deficiency. That's by far the most common type of anemia, but that could happen. Or it could be you have inadequate hemoglobin, like sickle cell. So in this picture here, you see a couple normal cells, right? Here. But then you see the edge of one and the edge of the other that are sickled. And what happens is sickle cell anemia is a point mutation. It means it's a single mutation of a single amino acid that causes the hemoglobin molecule, when it unloads oxygen, to make the red blood cell misshapen. It actually gives people with a single trait of that an advantage from surviving an attack of malaria. So you see it in people that have ancestry to parts of the world that tend to be near the equator, that have high incidence of malaria. Scientists have just at least traced at least 18 different independent events that led to mutations that cause the sickle cell gene to become in the population, again, because it gives an advantage to do that. But it does compromise your ability to carry oxygen and does cause other issues. When we describe anemia, we typically describe it using two things. One is by its cell size, and if we do that, we use the word cytic. So there are ways in which we can measure cell size. We're not going to worry about it in lecture. We'll worry about it a little bit in lab, where you look at the size and say, okay, is it normal or not? So if it's normal cell size, we call it normocytic, normo for normal, cytic for cell. If it's too small, it's microcytic. And if it's too big, it's macrocytic. So here are all of our normocytic cells. Here's all of our microcytic cells. And here's all of our macrocytic cells. So we have those different cell sizes there. But that's not the only thing that we worry about. We also worry about the amount of hemoglobin. The hemoglobin amount is called chromic. We use the word chromic because remember we talked about hemoglobin giving color. As the cytic portion of the cell size is usually a DNA-based problem, the chromic portion, the hemoglobin amount, is an RNA-based problem because it's a protein typically. And we have the same idea, although we use different words. So we use the root word chromic. So if it's normal chromic, if it has a normal amount of hemoglobin, right, it's called normochromic. If it's got too little hemoglobin, you can see these cells here, right, are whiter because remember, hemoglobin gives us color. And if we have hyperchromic, that's up here, and they're darker. So we can look, and here's all of our hyperchromic cells. Here's all of our normochromic cells. Here's all of our hypochromic cells. So if you look, we've got a way to describe different types of anemia. So the most common type of anemia is microcytic normochromic. And we'll, sorry, not normochromic, hypochromic, microcytic hypochromic. We'll look at the next slide and kind of see what I mean by that. Okay, so here are some of the more common types of anemia. The first and by far the most common is called microcytic hypochromic. So microcytic hypochromics, remember that was small cell, low amount of hemoglobin. It used to be called iron deficiency anemia, but now, it's often from iron deficiency, but it could be from other things like chronic infection, where your body sequesters iron in the liver, which helps fight infection. But just show how common it is. 20% of women have it, 3% of men, 20% of school kids. And here's the big one, half of pregnancies result in iron deficiency anemia. So again, that would be down here at this area right here. So small cells, right? Microcytic, and then little hemoglobin, hypochromic. So it can't carry as much oxygen. Not all of them are like that though. There's a type of anemia that's called macrocytic normochromic. So that would be large cells, but have normal amount of hemoglobin. So that would be this one right here. One of the more common ones is called pernicious anemia. Pernicious means it's going to cause death. So that's something to consider. And it's due to a vitamin B12 deficiency, often absorption issue where you don't make intrinsic factor. And so might as well just finish this all up at once on this slide. So variation in blood cell numbers can be normal physiological responses, or they could be pathological as well. You just have to kind of consider them. So we just talked about red blood cells a little bit. So here's our red blood cells right here. So here are disorders. We talked about too little red blood cells, or not enough hemoglobin, or not enough oxygen carrying capacity. That's called anemia. If you have too many red cells, that's called a myeloproliferative disease. Okay, so let me erase that so we can see myeloproliferative disease means we're making too many cells in the bone marrow. So myelo refers to bone marrow. Proliferative means proliferate. proliferation, too many things. The most common one of those is called polycythemia vera with red blood cells. So it's really not a great name because poly, many, cyte, cells, emia, blood, but it's just specific to red blood cells, not general. And it increases viscosity, as we talked about before. Increased viscosity of blood for athletes is okay because they can carry more oxygen if they have too many red cells, but bad for smokers because they're more likely to have a heart attack or some sort of things like that. For the white cells, the white blood cells, if you have too many, it's called leukocytosis. So leuko, again, white. Cytosis means excessive number of cells, basically. It's okay if you have an infection. As a matter of fact, a normal response to infection is to make more white cells. That's good because your body's going to be able to fight that infection off. But a abnormal response, a bad response, would be leukemia. Leukemia is when you have cancer in the body where you make too many of the cellular elements. In case of this, it would be white blood cells. Can come in two places. One is myeloid. Again, myeloid refers to bone marrow. So if you had a bone marrow disease, it would be myeloid. It could be lymphoid as well. So if we're talking about leukemia, leukemia is a cancer. Cancers invariably produce too many cells. So if you had lymphoid leukemia, that means that the problem was in the lymph. And so you'd have too many of the white cells that are produced in the lymph. That would be the monocytes or the lymphocytes. So that's leukocytosis. On the other side is leukopenia. Leuko-white, penia means too few. And leukopenia is always pathological. It's never good not to have as many white blood cells as you should. And it means you're immunocompromised, that you're not going to be able to fight off infection as easily because of that. So leukopenia is always pathological. Leukocytosis, though, as I said before, sometimes good, sometimes bad, depending on why that happens. Platelets will be the other one. So platelets, again, going back to the scientific name, if you have too many, it's called thrombocytosis. So you see the cytosis word, right? Too many. And then all we have to do is put the scientific name for the two cells, leuko or thrombo, in front of it. Thrombocytosis is good if you have an injury because you won't bleed out, right? But it's bad because it causes thrombosis. Thrombosis is just another scientific name basically for a thrombus. That would be a blood clot, right? So a stationary clot in an unbroken blood vessel is thrombosis. Going the other way, if you didn't have enough of your platelets, you'd have thrombopenia. Again, penia, right, meaning too few, and just put the scientific name in front of it. That's always bad because you'll have a decreased clotting and a greater increase of bleeding out. And as an example of that would be somebody with like hemophilia. So those are the blood disorders that you're responsible for this class.